Right now, inside your body, a suite of molecular machines is copying all 6.4 billion letters of your DNA genome to create an identical copy every single time a cell divides, and it has to get almost every single one right, or you could get cancer. DNA is a twisted ladder. Two long strands, each rung is a paired chemical base. A always pairs with T, and C always pairs with G. That pairing rule is everything. To copy itself, the ladder has to split down the middle. An enzyme called helicase acts like a zipper pull, unzipping the two strands apart. In human cells, each replication fork moves at around 50 to 100 base pairs per second. But here's the trick. Thousands of forks open up simultaneously across the genome, so the whole job still gets done in hours. Now each exposed strand becomes a template. DNA polymerase, the actual copying machine, races along, reading each letter and snapping in the matching partner. A gets a T. Think of it like a zip file copy, but instead of bytes, you have 6.4 billion chemical letters being duplicated in just a few hours, thanks to thousands of molecular machines working in parallel. One cell becomes two, each with a full copy. But here's the curiosity gap. Before proofreading, polymerase makes roughly one mistake per 100,000 letters. Across 6.4 billion letters, that's about 64,000 raw errors per copy. So how are you still alive? Polymerase has a built-in proofreader, like a spell checker that runs immediately. The moment it inserts a wrong letter, it detects the mismatch, snips it out, and tries again. This alone cuts errors by 99%. After all repair systems have done their work, the final error rate drops to roughly one mistake per billion letters. Then, a second crew, mismatch repair proteins, scan the freshly copied strand, looking for remaining warped rungs in the ladder. They patch another 99% of what slipped through. The final result? Somewhere between 1 and 6 errors per genome copy. Most of these mutations are neutral, having no discernible effect on health or fitness, because they occur in regions without critical known functions. However, some non-coding regions are vital for gene regulation or produce functional non-coding RNAs, and mutations in these areas could still have an impact.